So if our speakers could come have a seat. It feels a little bit like a firing range up here, I have to say. We spent some time last week kind of trying to come up with some provocative questions, and those are on the slide that Victoria has put up here. If you feel stymied by coming up with a question of your own, or you'd like to know the answer to one of these, which of course we don't know the answer to, but we can talk about what the answer might be. So.
Hi, I have a question for Tatiana, which is, what about ballast water? Could that explain your biogeographic migrations rather than ocean circulation?
Yes. Yes, it could. I would love to talk to you about it more.
Hi. So I think all of you have touched on this question, so I apologize if I'm repeating what all of you have sort of said, but I think it introduces you something about the experimental evolution experiments not sure realistic. And I run numerical models, and I run them into the future, and like all the reviewers always say, but you don't have adaptation in your model. So yes, okay, good point. But for instance, let's just think about an example of a warmer species taking over the space of a cooler species because the water is warmed up. The time scale for which an evolution could happen versus the time scale for a competitor, a warmer water species, to move into the cooler species is, I guess, the crucial piece. If it's quicker for a competitor to arrive than something can compete, then the evolution doesn't matter because you've been outcompeted already. And so you've all touched on this at some point, but I think it's kind of crucial to the ocean. And in some ways, you'd said that, Elisa, you had said evolution in a lake and an evolution in the ocean are similar. But in fact, in the ocean, that piece is particularly important. The fact that in the lake, everything is somewhat contained and we assume well mixed, but in the ocean, you could have this competitor move in, and no matter how much you've evolved, it doesn't matter. You've been outcompeted before you could evolve. And so you'll probably have a comment on that.
I'll mention one thing before I turn it over to some of these guys. And there's an interesting paper by a group of marine biologists a couple of years ago. I think the lead author was Simons. It was a science paper. And what they did is they looked at this idea of changing biogeographies of different organisms and warm water species taking over where cold water species just can't hack it. And in their modeling runs, one of the things they point out, and I think is relevant for this audience, is that they come up with something called no analog communities. And those are communities of organisms in their model that coexist together in the model that in no place in today's ocean coexist together. And so they were talking about the potential for new kinds of interactions. They didn't talk about biogeochemistry. They were dealing mostly with sort of marine biology. But I think that may be happening, where we may have these no analog communities. And even when we do get no analog communities, there's evolution happening there. And something that we've all talked about in our sort of preliminary conversations to this session, but it didn't actually come out in any of our talks, was that evolution's always happening, right? So organisms are literally running to stand still. So even if we don't see a change in any kind of phenotype, there's a lot of movement under the hood, so to say. So it's hard to envision even that organism that's taking over is also evolving as it's taking over. That makes sense.
Yeah, I would just say that one thing you got to think about is just the gigantic population sizes of some of these things. So if you take Prochlorococcus, based on the size of their genome and the number of cells there are, every possible codon change of every protein in their genome already exists. Actually, it arises every day somewhere on the planet, right? So whatever mutant could give you an advantage already exists right now somewhere. So the question about the pace of evolution is how big of a fitness advantage do you have? And the more extreme the environmental change, the more of an impact evolution would have as opposed to migration, I would think about. So the faster the environment changes, the more you have to think about evolution.
Hi. I have a question. Well, half of the question is a little bit technical for Victoria, and the rest of the question is a little bit philosophical for the whole panel. The technical question for Victoria is, how do you control your model so that you don't have a superbug that rise up? And the philosophical question is, let's say if truly, as you said, the microbes are just fitting into the niche, right? Like you're selecting that there are different microbes that can fit into the niche as long as they have the same function. So if that's the case, if there is a superbug that has that super function, wouldn't it take over? But why is that not emerging in the nature?
Great question. So I think I have a mic, so you can have that one. So this is a huge problem and challenge and concern in the model, because the observational evidence that carrying genetic complexity is something that should be penalized is kind of mixed. So dinoflagellates have this tremendously diverse genome, and they seem to do fine, versus we see streamlining in other organisms that we attribute to the fact that they live in very tough environments, for example. So in our model, we penalize complexity essentially through reducing growth rate as just one of the possible combinations. And we tune that complexity to ensure that we get organisms that still carry diverse number of genes, as well as streamlined organisms, and that all of the genes in our model are represented somewhere in the domain. So those are kind of some of the criteria that we use to set those penalties, but they're totally ad hoc, and we need to know more about that. So philosophically.
I don't know. I think there are certain times when superbugs are present. You can think of some of these massive phytoplankton blooms that are often dominated by a single species. They're superbugs for a short time, and then the environment changes. So we have a really dynamic ocean environment where in some cases, yeah, we do have organisms that really tend to dominate. But we also have a lot of diversity out there, and I think that's that interaction term that Jeff talked about between organisms. There's the environment that's changing, and then there's stochasticity on top of it.
And viruses that change faster than anything else, right?
Yeah. And oceans are basically virus soup, right? There's more viruses. And oceans are basically virus soup. There's more viruses in the ocean than any other kind of organism. That's
12 million whales worth, I hear. Mega whales.
I'm glad you invited me, and I just want to thank you for an extremely thought-provoking session. And I'll tell you why. I just returned from the Antarctic at Pete Conway and I for a project there where we were looking at phytoplankton microbiome effects affecting each other from the perspective of phytogeographical cycling. And we took the approach that one of you said, you ought to do continuous projects, which is what we did. So we have a continuous system for whole community in which it was controlled by one factor. It doesn't matter what it is. But you just made me realize that there's probably all of these other things that apply to it as well. I can tell you that the communities absolutely reached stable state in a matter of weeks. We presented this at the Ocean Sciences meeting, and so we ran this experiment somewhere a week to 2 weeks long. This last time we ran it over almost 2 months, changing the conditions. And so my question to you is, some of you were strictly on small scales, 300 generations, which is still in the days or whatever of a culture. And then others know I read over time, decade of time scales. But all of a sudden, I have data that shows that they reach whole communities, stable conditions in weeks to months. So is that evolution, or what is it? I just want to be enlightened.
I think a lot of it could be to do with rapid sorting of what you had in your community to begin with, right? Because you're going to be putting in a really mixed community, and that is going to be sorted really, really quickly into whatever stable state it reaches. So I'd say it's probably ecology to begin with, and then evolution later. I would say maybe you have opinions on that also.
Yeah, I would agree with that. I would also say that probably what you're seeing is sort of the result of like co-evolved interactions within your starting community, right? So you're seeing sort of like the core microbiome of that, whatever that community is. So the things that were there that kind of drop out are sort of either transient or they're dependent on something that's no longer present in your culture condition. And what you're seeing in that stable community is going to be this network of things that depend on each other for whatever reason.
I want to follow up on Jeff's comment, and that is, so ecology and evolution are really intimately linked. And so sometimes it's really hard to tease apart some of those activities. And we've tried as disciplines to split them apart. And probably one of the most exciting new areas for ecology and evolution is this area of looking at eco-evolutionary dynamics and looking at those feedback loops that are happening between ecology and evolution. And those kinds of experiments, when they're sampled in particular ways, can kind of tease apart those feedbacks. But yeah, you probably have a combo of both evolution and ecology happening that leads you to this new stable state. And those kinds of experiments are also really interesting because they can give us a sense of the timescales that these shifts happen on, and then how long do they, how long does it take to get to something stable? So it's super interesting.
I had a question for our panel. We've spent a lot of time talking about sort of this redundancy in function. I think really kind of, Victoria, you really drove that point home. And I'm thinking about particularly microbes where that function's not terribly redundant and there's not a lot of genetic diversity. And some of our really important microbial nitrogen fixers fall into that category, where John Zare's group has shown in a kind of globally widespread single-celled nitrogen fixer that there's not a lot of genetic diversity. And there's a lot of potential for nitrogen fixation in the environment, but the taxa that are doing that are fairly rare. And so I'm wondering, maybe starting with you, Victoria, did you see those kinds of taxa in your model, and how are you thinking about those as opposed to photosynthesis or something that's more ubiquitous?
Yeah, of course, we've been interested in nitrogen fixation in different groups of organisms that do that for a long time, and it turned out, as we sort of expected, that in this model, without a strong phosphorus constraint, it was pretty hard to rein in the nitrogen fixers, and so I don't think that their role in the community was realistically represented. But the issue, I think, is really interesting because it kind of touches on, we saw some thermal curve response curves, for example, for different organisms, and learning to understand the genetic underpinnings that drive those response curves is, I think, essential for doing the kind of modeling that I'm interested in doing. So rather than just knowing that an organism has a particular curve, understanding what are the protein sensitivities, or what is setting those limits and how is that changing and how could that change is kind of, so we really need more mechanistic information about what's driving those changes to model them well. Otherwise, we're just randomly setting, response curves that we don't know how to adapt. I don't know if that really answers your question, but maybe you guys. I mean, some of the experiments you were involved in with the community temperature response, these are really important to start to look at the mechanisms that are setting those changes and differences.
Am I up? Oh. So I was involved in a project for about 10 years in the Gulf of Mexico where we were trying to figure out how we could get enough nitrogen into Karenia brevis cells to have these massive blooms. And we went through all these contortions and all these permutations and measured every rate we could think of and all the off-land runoff and upwelling. And in the end, it came down to physics. And so I wanted to follow up with Victoria on if there is this infinite level of plasticity out there and everything's pretty much exists everywhere, does it just come down to physics?
I raised that already, so I'll let my colleagues have their time.
No. Well, right. Okay, I'm just trying to figure out the best way to say it. So, I mean, physics will control some things, right? I mean, it'll control the size of the community that you can support, right? But there's other things that are important in there as well. I mean, so sort of like what forms do the different elements take in there, right? I mean, the balance between like ammonium and nitrate or carbon export. And I think all of those things are going to depend on other things besides physics, right? The particular interactions between the different functional groups of phytoplankton. I mean, I don't know. I don't have like empirical evidence to back me up, so I'm just like waving my hands here. But I do feel like there's other things besides.
And I wouldn't be able to go home if I didn't mention that there were grazers out there. That I see some people cheering out there. And so, of course, right, so the transformations that those grazers are mediating and where they're doing that and when they're doing that is separate from just the physics. And I think that plays a large role in moving nutrients around in a way that is then mediated by the physics, but the grazers are playing a big role there. I think I can go home now.
But ultimately, isn't everything physics? I mean, biology is just physics.
So my question or comment is on the same line. So if evolution is non-deterministic in the ocean, so that you had an alternate universe, but you get the same biogeochemistry with what evolved, that's on the presumption that all niches are currently occupied. So I can think of a world, say, where calcification never evolved, and that would change biogeochemistry radically, or silicification didn't evolve, or things like that. So based on what you presented, what are your comments? It's more of a comment than a question.
Well, that's a really heavy philosophical question. It's one that is fun to think about. I mean, you can look at the fossil record, for instance, and see this succession of reforming organisms over, the eons of time. And there's always been reefs, but they've been made of different materials at different times in the past. So, I mean, there's, you can convince yourself on one level that evolution is totally deterministic because it keeps producing things that we can say are similar. But on the other hand, is a reef made of silicon fundamentally different than a reef made of calcium carbonate? I mean, I don't know. I mean, they're fun things to think about. It's really hard to say.
Well, I hope I expressed that, that evolution of the library of metabolic function is pretty key to our model. All of those pieces needed to be in place. So that is not a non-deterministic function. So we were taking a shorter term approach, you might say.
Ooh, I can.
That might be helpful.
Okay, so an adaptive landscape. Oh, gosh, how to explain? You put me on the spot. So basically, what you can picture an adaptive landscape is as a landscape that has peaks and it has cliffs and organisms. Depending on what kind of phenotypic traits they display, they sit in different parts on this adaptive landscapes, and they might not sit on the peak, but they might sit near a peak. So, and evolution is actually almost never optimal. So you're almost never going to be sitting on that peak of being optimal. Otherwise, all of our plants would be black because black is a much better color to absorb sunlight, right? But we don't really see that. So for most organisms, it actually kind of makes sense to be a little bit moved from that peak, especially then if the environment changes, that you have a little bit of leeway, so to say. So I'm thinking, if you imagine a world, yeah, I've kind of lost myself here a little bit.
And is that, in addition to that, there's this idea that adaptive landscapes may have multiple peaks, and where an organism sits may not actually be on the highest peak, but there's a great valley between in terms of a fitness valley, right? You have to have a decrease in fitness to get to that other peak. And so often what we're seeing, and plants not being black might be one of them, that getting to that other peak is very difficult. And so we do see some constraints on evolution, and that you're not always exactly at your optimized spot in either physiology or habitat.
The question
we have had a hand up for a long time.
The question is, how do you account for, in your model, for epigenetic changes, or can you?
So I'm not sure that there's an important difference in a model between epigenetics and plasticity, right? So epigenetics is kind of like plasticity that gets sort of like hardwired between generations. But if you're kind of modeling things as pools of elements inside of a functional group, I don't know that they would come out differently, right? So that's kind of how I would answer that. I'm not a modeler.
Yeah, I mean, we've talked about different ways to approach the replacement of organisms, whether, rather than throwing a brand new organism in, maybe you just tweak one tiny element, one gene, or one aspect of the fitness of the organism that died out. But I think we've sampled so little of the diversity in our model as it is that we didn't feel that that was necessarily productive yet, because we hadn't really sampled enough of the potential range of organisms with the 79 that we had in the model at one time.
Hi there. This idea of what's the ultimate control of physics versus other processes is really interesting. And another way to think of it perhaps is that the reactions that are being conducted by these organisms are essentially enzymes that are catalysts and have ultimately a thermodynamic control that's controlling their reaction rates. And that's sort of an interesting contrast. And I wonder how, for example, in Victoria, in your model, how are you accounting for sort of that chemical space in terms of what are you allowing these organisms to evolve to? Is there some stopping mechanism to prevent them from evolving to something that's too efficient than chemistry can actually allow?
No, and I think it's I was trying to allude to this to some extent in talking about the temperature tolerances of organisms. In fact, we completely avoided giving any temperature tolerances on any of our organisms, because in practice it allows you to set the distribution of organisms I know what the temperature is going to be in my model in a given location, so if I say the bug does really well there, that's where he's going to be. And we wanted to have a more flexible ecosystem, and we knew that we couldn't have enough organisms to assign each one an infinite number of thermal tolerances. So I think getting at the mechanistic underpinnings of what drives those and attacking that from the genetic perspective makes a ton of sense. And trying to understand what is it that makes some organisms have really wide thermal niches. Why is it that we seem to see a lot of organisms backing up on that 30-degree threshold? Is it a fundamental physiological constraint, or is it just that that's what we see in the ocean today? Those are questions that really need to be better understood. Maybe you guys have.
We'll do one more question, and then I think we'll break for lunch.
Thank you. So I'm trying to reconcile a little bit something that I heard both in your talk, Jeff, and yours, Tatiana, and that is, so Jeff, you were talking about how structure allows mutualisms to occur, and you gave the example of coral reefs and their symbionts, and that in the ocean there's not that kind of structure. And then Tatiana, you were talking about these bacteria populations that match onto diatom populations in very different parts of the world's oceans. And so I'm just trying to reconcile, maybe I'm not thinking about it right, but does the ocean have a lot more structure than we think it does that allows those relationships to happen? And sort of enter into some of these sorts of these patterns that we're seeing.
So, okay, I'm not sure if all the bacteria that Tatiana was talking about are physically attached to the diatoms. So that's kind of an open question, is how much time do diatom-associated bacteria spend actually touching them versus swimming around close to them, right? I mean, well, like this fundamental physical problem that you have if you want to be planktonic is that if you get too big, you sink, right? So if you're a bacterium and you want to keep mooching off of a photosynthetic cell, you need to make sure it stays around the sun. So if you attach to it, then that might be a problem because number one, you're blocking light, and number two, you're making it heavy and it starts to sink. So there's this incentive, I think, to be close and not actually attach, right? But at the same time, you're also pressured to get closer to it than your competitors who are also trying to compete over the same things. There's probably some kind of equilibrium there. Regardless, I don't think that they're going to end up in these structured, dense populations like you associate with the inside of an animal, right? Or with a biofilm attached to a surface. And that's where you're more likely to see kin selection playing out in the evolution of real mutualisms. So. And I think, granted, we don't know how much physical structure there is in the plankton, but I think that sinking problem sets some limits to how much there can be and how sort of long-lasting it can be.
But there could be some kind of chrome sensing, no? So they could be near but still feel that the bacteria are near or that the...
Yeah, so they communicate through chemical signals, right? And we know that diatoms do this. Diatoms and bacteria communicate back and forth through diffusible chemical signals. So yeah.
And I think there are clear examples of that structure. You can think about diatom-diazotroph interactions, right? Perhaps the diatoms, the bacteria-associated diatoms, and there's probably a strategy that bacteria are trying to optimize there. Sticking to or sticking close to some approximation of a structure if they're not an intracellular or physically attached. They're optimizing that sticking close to versus having some cells being free-floating to get to that next structure. I saw Uta was out in the break, Uta Passo, and so she thinks a lot about this marine snow, right? And similarly, that provides a structure, but that structure is inherently, it's sinking, and so the bacteria are taking a ride to the bottom. So there must be some strategy for either offspring to then become free-floating or to spend a certain amount of time on a structure and then head out.
Okay, I think we have to end the session for now. Let's give another round of applause to our amazing speakers.